Very happy to be here this evening, kind of expand on what Bob has set up in terms of the DNA barcoding project that has been undertaken by the University of Guelph, and really talk about kind of where my research interests lie, which is how do we move beyond just DNA barcoding and use this amazing resource to do some other types of testing that get beyond some of the limitations of the straight sequencing method that is often used. So my presentation points are going to really cover how are we translating this biodiversity science into something that can actually be used by people who would benefit from doing this kind of testing. So really focused around industry testing and bringing this into the market where it could actually make a difference. So I'm going to start with talking about how do we read this library and move from these huge numbers of sequences to kind of the Coles Notes version of looking for very specific things within the library, and moving to how do we achieve on-site species identification for cases where you need to have an answer really quickly, which is often the case for some of the food and environmental applications that I'm going to be talking about, and two different case studies to go over what we've actually been using this technology for. So this is a screenshot of the bold systems, which is kind of difficult to see, but the point of this really is just that you have millions and millions and millions of sequences that are in these different libraries. And this is very overwhelming for an industry person to be able to have to go in and make matches on. Going into a publication that is in the scientific literature is beyond the capacity for most regulators or industry. Being able to pull out a method from a scientific paper is not something that most people are going to want to or potentially be able to do. So how do we make this something that they might actually be able to use? Well, what we can do is make use of the amazing database that's been put together in BOLD and these vouchered sequences that we know come from expertly identified materials, and then use those sequences to compare that longer sequence and look for portions that are specific to a certain target. So instead of doing a Sanger sequencing method like DNA barcoding that's going to be looking for a larger sequence, we can focus in on a couple of key regions that tell a certain target apart from all of the other ones. So what I have here is the illustrative DNA barcode, so that full kind of colorful thing of ATs, Gs, and Cs there. And in the middle is a portion that we would target for the type of testing that I'm going to be talking about, which is real-time PCR. And this is one of the kinds of DNA testing that can be built from libraries like the DNA barcode library that's been put together, that gets around some of the issues that were mentioned before with degraded DNA, being able to identify mixtures, and most importantly, portable testing, which I'm going to be talking about pretty extensively in the case studies. So really what we're able to do with this type of test development is take the reference library sequences or DNA barcodes, look at portions of that DNA barcode that are targeted to identify a specific species. So we might be using 3 different regions there, so the little arrows in the middle and the P, that are specific to Atlantic salmon, for example, and that we find those portions within the larger barcode compared to all of the other salmon sequences that are in that library. And then we can use those species-specific primers and a fluorescent probe in the middle of them to actually monitor that copying of DNA in real time, which means we're cutting down on the steps that are required to actually get our answer. So instead of having to do the DNA sequencing and compare it to a library, the process actually stops at that PCR amplification stage, and the fluorescence that's generated tells us whether that target is there or not there. So you have this really nice simple output of it's either there or not, so you have a yes or no answer for your target, and it takes a lot of the time and cost out of actually processing the sample. So this means that we can actually make use of the DNA barcode library for cases where an answer needs to come back that's time sensitive. So if you have a shipment of a perishable commodity at a border or at a warehouse, you kind of need an answer quicker than a couple of days or a couple of weeks that you might have with sending a sequence out to a laboratory. You can look at degraded DNA, so we can do this on pretty much any type of processed food product, and also bring this into the field. So this is kind of one of the innovations that has come out of real-time PCR space. And in general, this method's been around for a couple of decades, mostly focused around human health and pathogen detection. And along with that, some portable systems have been made to be able to do that human health identification in the field. What we've been doing is capitalizing on the method and now on the new equipment that's available to actually be able to do this in other applications as well as human health. So this is a kind of array of some of the different portable technologies that are available. So they're anywhere from a laptop and kind of benchtop box type approach all the way through to this Spartan Cube, which is the new one that's 4 inches by 4 inches cubed. I'm going to focus on one of the ones that we've been working with to date, but we've used several of these different methods. And really what it comes down to is that we can take this library of DNA sequences, make targeted testing for specific species to be able to do the on-site identification with a simple method and a portable instrument. And this makes testing a lot more accessible to a lot more different kinds of people in a lot more different kinds of places than you'd be able to do with sending sequences or samples rather out to a laboratory. So instead of having an answer in a couple of days or weeks, depending how busy the lab is, you can get an answer yourself in less than 2 hours. So this is the instrument that we have been using, and the key also is that we're working on a sample prep, so DNA extraction protocol that's actually totally lab-free with this company. So this is all of the equipment that you need to extract DNA, run your sample, actually get a test result back. So I think afterwards we can kind of open this and go through it if anybody has questions, but that's what we're working with right now as far as what we're trying to make commercially available to industry. And the reason for that is that right now the landscape for food fraud in particular is kind of messy. There is a lot of media coverage around different instances of substitution that are being found. You have headlines like delicious or deceptive, honest players need to step up, and some recent prosecutions for food fraud both in the US and in Canada. And one of the issues is for industry is how do they actually get ahead of this and be able to do something about this if they're doing something that's actually good, which I think a lot of suppliers are. So I'm going to focus on seafood in particular because it's such an important issue. And currently, there are 146 million tons of seafood harvested every year. This represents a quarter of the entire global protein intake. And additionally to that, 1/10 of the population of the world relies on seafood production, whether it's wild caught or aquaculture, for their economic income. So it's something that is critical to a lot of people around the world, both for food and for their livelihood. And yet 90% right now of fisheries are either fully or overexploited. So we need to be really careful about making sure that we're managing fisheries in a sustainable way, and species identification is one of the ways that we can help manage that. The other thing is that seafood has an incredibly complicated global supply chain, like a lot of products that are out in the food market now. And most of your products that you're purchasing have gone through multiple countries with multiple different regulations in each of those countries, possibly multiple different processing steps. And oftentimes the requirements around those products are traceability of, you have to know where that product came from and where it's going to, but beyond that there are no regulations around what you need to know about your product. And so you have this concept of traceability. It's required in a very narrow way, but also there are not really authenticity requirements. So even if you could trace that product back, nobody ever tested it at some point in the supply chain to say we're tracking the right thing. So we're trying to change that, but it is a difficult thing to do. So in the United States, there are 1,800 different species that are listed on the FDA seafood list. So it's not something that is easy to do. There are a lot of commercial fish species. It's not something that a regulator is going to be able to do. Even if you had the whole fish to deal with, you couldn't possibly visually identify that many species. However, species identification is a really important thing for seafood because the regulations, as we kind of already talked about, relate to the actual species that are allowed to be marketed as certain common names. So from a regulatory perspective, you should be able to identify your species. And we've already talked about the health, economic, and conservation impacts from substitutions in this space. So really, species mislabeling in seafood is a threat to consumers because you're losing possibly money, possibly there's health implications, possibly just losing out on that choice to be able to say this is the type of food that I want to be eating and I want to be eating in a sustainable way. And it's also a threat to businesses because there can be issues when media articles come out on seafood is mislabeled without really being able to address the problem. And finally, sustainability of that seafood harvest and production that's so critical. So what we've been trying to develop is some rapid and simple, which is very key, testing to be able to support industry who wants to be proactive about doing this type of testing to protect their supply chain. So I'm going to focus on one of the tests that we've developed, which is for Atlantic salmon. So this is a screening test that will be able to tell you if your fillet or piece of sushi is Atlantic salmon or not. And this is a really interesting case because oftentimes DNA barcoding can't tell you whether your fish is wild or farmed because it often is the same species. But in the case of Atlantic salmon, there aren't really wild Atlantic salmon anymore. So if you've got an Atlantic salmon, it's probably farmed. And there are some concerns around farmed fish that people may have around the dyes or antibiotics that might be used. The fatty acid composition from a health perspective can be different between wild caught and farm species. And the big thing is that they cost very, very different. So you might be paying a third of the price for a farmed Atlantic salmon as you would for a wild sockeye fillet. So to develop the on-site testing, basically we've worked with a company based out of Philadelphia called BioMeme, and we are working with them on optimizing a field test for doing seafood testing. So basically you have a field sample prep kit. No other equipment is required, so there's no heating, no centrifuge. You take a swab of your fillet, add it to a buffer. There is a syringe that you use to wash through a few different buffers that are shown here in the different colors, and the last tube actually contains your DNA. So this takes about 5 minutes. You add that to freeze-dried reagents that have everything you need to actually run the test. So everything kind of comes easy to use. It can be stored at room temperature. It takes a really, really short amount of time. So this is compared to what we would probably do in a lab for DNA extraction that would take a few hours, half a day sometimes, depending on the sample and protocols that we're using. So really, really simplifying the protocols for sample preparation. And then the second step of the process is actually running the test, and that's where this machine comes in. So this is actually the thermocycler that runs the real-time PCR. The freeze-dried reagents that have your DNA in it go right in the top here, and then you can see there's an iPhone in the front there, and everything is run on an app. So it's something that's really accessible in the sense that people are used to using apps for things. So it's something that you don't need to have a PhD in molecular biology to run, and in fact, pretty much anybody can do this with really minimal training. So you just add your reagents to the instruments, select your protocol from a drop-down list in the app, and then run it and review those results. So it takes 60 to 70 minutes for that part to happen. So you have about an hour and a half basically to wait from when you've actually sampled your product to when you can actually make a decision. For example, are you going to accept this shipment of fish that's just come in or not? And it's a little bit hard to see on the screen here, but this is a graph of, maybe it's easier on the other one, a little bit. So you can kind of see the curves that are showing up there. Basically, that's an indication that there's a positive sample, that there's some amplification happening. That just means that that fluorescent probe that I was talking about is actually amplifying something. If it was negative, it would be completely flat. So it's really simple to interpret whether you have your Atlantic salmon or not. The other thing that this instrument is set up to do is actually upload everything to a cloud web portal, so results can be tracked from anywhere in the world with a password. So if you had a network of different machines at different places, you can access those and track what testing is happening, who's running the testing, and what those results are from anywhere. So we've done some on-site testing with this at a large food distribution center. So we did this with actual warehouse employees, so minimal training, about 20 minutes on how to actually run the test and what you're actually what you're actually doing in the different steps. And we did almost 70 different samples with 100% success across the board. So all of the ones that were supposed to be Atlantic salmon came back Atlantic salmon, and all of the other species were negative. So in terms of commercial testing, we know that real-time PCR, through our different testing in the lab and also now beta testing some on-site approaches, is useful for seafood ID. It's rapid, it can be really portable, and very, very simple. And what we're hoping is that this improved access to testing will improve authenticity for industry or regulators that want to be doing a better job of actually enforcing species identification. And that this is going to augment some of the traceability schemes that are already in place for regulations to improve the sustainability of the industry as a whole. So we've worked out now on kits for Atlantic salmon, coho, chinook, sockeye salmon, swordfish, mahi-mahi, and blue crab, and that's just our seafood tests. And the tests actually feed into a certification program through a company called TrueID that actually spun out from the University of Guelph. So this is another thing that we're doing to try to translate some of the knowledge that is coming out of the university to actually be able to do that improvement in actual industry. So we have several clients that are actually certified. So Jameson products and Nature's Way products actually have a little TrueID check on it that has gone through our DNA testing for authenticity. And this is something that businesses can actually use to communicate to customers, we're doing this additional level of testing, protect their brand by actually doing that testing. And we have this registered trademark for DNA authenticity. So the second case study that I want to go through is similar in the sense that we're still actually using this instrument, but we're adding another level to the testing by doing some environmental monitoring. So this all centers around detecting environmental DNA. So I'll give a quick breakdown on what that is, but basically it's any genetic material that's obtained directly from an environmental sample. So it might be soil or water or sediment or anything like that, but there's not actually signs of that biological source material. So instead of taking a piece of fish or frog, in this case, up on the screen, what we are able to actually do is from tissues and cells that are actually shed from the organism, you can detect that that organism has been recently in an area without actually being able to find it. So you can use this to do environmental assessments for things that might be really rare in an area without actually having to trudge through and turn over every rock to try to find that organism. So environmental monitoring in general is really any technique that's used to characterize or monitor the environment. And traditionally, it's been done by visual identification, which can be very expensive because it's very, very time consuming. So there needs to be a lot of time spent in the field trying to find the things that you're looking for. But this is something that's required for assessments of environmental impact for different areas. So before a new building or plant would be built, you need to do an environmental impact assessment to see, are there any endangered species around? Is there anything that's commercially important that might be impacted by this new development? So very, very important, but very expensive and time-consuming process. So environmental DNA has been proposed to sort of revolutionize how this environmental monitoring is being done. However, it's still very time-consuming, not as compared to the visual identification, but there's still quite a lot of lab work that's required, and there's a lot of risk of contamination because you have to bring samples in from the field. You have to do a lot of decontamination of the lab, also all of the equipment that goes out into the field. So what we're working on to solve this is on-site solutions where we can actually do environmental DNA detection, which is much more rapid and lower cost than some of these other methods. So in addition to the DNA extraction and real-time PCR steps that I've already talked about, we also needed to figure out how we're going to be able to automate water sampling on site. So in this case, we're focusing on aquatic environmental DNA. So what we need to be able to do is take a large volume of water and filter out the actual DNA onto a piece of filter paper. So this is something that you would traditionally do by taking liters of water out, taking it to a lab, using a centrifuge to filter it down. What we've actually done is work with a company who traditionally had been developing backpacks for electrofishing, and they have now developed a backpack to actually suck water out and filter it. So we've combined this additional step for on-site water filtration with our on-site DNA extractions and real-time PCR to be able to actually test for environmental DNA right on site in the field. So one of the examples I'll go through of where we've actually applied this is for Jefferson salamander. So this has been declared endangered both in Ontario and on the federal list. It consists of the actual Jefferson salamander, but also is a complex of other species. So you have the Jefferson salamander, which is actually protected, but also the blue-spotted salamander, so A. laterale, and then a hybrid of the two. So the hybrid and A. laterale are not protected. However, they all look pretty much the same. So you need to use genetic methods to separate them out, and generally this has been a lab-based approach where you have to send tissues to a lab and wait to see what's actually, what actually have you got in that space. And they all kind of co-occur in the same areas. So what we were able to do is go to 9 different pools, some of which had historically tested positive for Jefferson salamander and some that had tested negative. Take our water samples using our new approach, extract our DNA on site, and we went at 3 different times of the year to do this. So basically what we wanted to do is test how does our method compare to what's already being done for visual assessments? Are we getting the same answer as what's traditionally being done? And we found out that yes, we are. So we actually got 100% almost correlation with what the historical data was showing in all of the different sites that we were doing. And this is something that takes a fraction of the time and cost to the traditional assessments that were being done for this species. And we can also do this for egg sacs that have been found that you wouldn't be able to do morphological identification of. We can take those eggs that have been hatched and identify whether they're from Jefferson salamander as well. So you can use this at any of those life stages to identify what species we're actually finding. So this is another spin-out company from the University of Guelph that's been developed to actually address this problem for companies that are doing environmental assessments who may not want to have a molecular biologist on staff, but may want to actually be able to benefit from environmental DNA testing. So what this company can offer is time and cost efficiency savings. So again, less than 2 hours from sample to result for these targeted species. We're working on about a dozen or so species that we have tests for now, and that roster continues to grow. We have a field-ready kit for doing all of the analysis through from water sampling to answer. And we can use this to do detection and mapping of species at risk for early detection of invasive species. So if there's concern that an invasive species might be spreading into an area, we can proactively test to see has it actually gotten into the area yet. And looking for keystone and indicator species for water quality assessments as well. So I'll just wrap up by saying thank you and some of the acknowledgements of people who've worked on this work. And this is I guess the photo of our textbook on seafood authenticity. And I guess we have time for questions now. So.